Hello, everyone. This is Phil Cardoso from the University of Illinois, and I have a guest today to talk about her most recently published paper, an article in press, Dr. Denicol. So maybe we can start, Anna, you can introduce yourself and what's your position at UC Davis, and maybe you can tell a little bit of your story. How did you get there?
Well, first of all, thank you so much, Phil, for inviting me. This is really great. I watched previous episodes of your podcast, and I think that's really a great idea to be able and really great opportunity for us to be talking about our recent paper. So thanks for that. And so my position here at Davis is I am an assistant professor, and I am at the Department of Animal Science. Been here for 6 years now. And mostly what my lab studies is related to reproductive biology. So you will say, well, the SLICK is not really related to reproductive biology, right? Which is partially true, but also I think that really important is an effect of the environment on reproductive biology, right? And that's something that I've always been interested in, which is how does heat stress impact reproduction? And of course, I'm not by any means the first to study this. There is a lot of work, really good work out there, but I have always been interested in not only understanding how heat stress may affect reproduction and, of course, other parameters of a cow production and well-being, but also ways to try to get around the negative effects of heat stress. And that's where the SLICK mutation comes along. It's something that Peter Hansen, my PhD major professor, was very involved and is very involved with. And so I've known about the SLICK allele of the prolactin receptor for a long time. And when I had the chance, we also started some projects here in my lab to study the effects of this mutation in Holsteins.
Okay. And so we're going to talk about your paper on the SLICK1 allele and a little bit of background. How did you get to the SLICK1? Why not SLICK2? I don't even know if it exists, but like, why not other alleles? How did you figure out that this is something that we may be looking into it?
Yeah, so again, this wasn't mine, right? I'm not going to claim ownership of this. Of course, there's people studying these mutations for a long time now. And the SLICK1 is called SLICK1 allele because it was the first one to be described. And there are other alleles, SLICK2 today, there is SLICK2 to SLICK6, so SLICK1 to SLICK6 really, which are different mutations in the prolactin receptor gene. And so all of these alleles, they mean different mutations that change the prolactin receptor a little bit. And so they form, for the most part, shorter versions of the receptor. And what happens there is, so talking specifically about the SLICK1 allele, is that there is a shortage of 120 amino acids in the intracellular portion of the receptor. And what that may do, which we are still investigating and trying to understand better how that changes signaling, is exactly that, right? Change the way prolactin signals in the cell that has the receptor. And so another question then becomes, well, but what's the relationship of prolactin with heat stress, right, or thermoregulation? And the answer is prolactin actually has important roles in thermoregulation. And so when we think about prolactin, I was just yesterday teaching my class and talking about that a little bit, just introducing the topic to my students. And it's the fact that prolactin is known for its main effects on lactation, right, on promoting milk synthesis. But we know now that prolactin has a lot of other effects in different biological systems in the body. One of them being thermoregulation. Prolactin has important immunoregulatory roles as well, so a very important role in the immune system. And in the skin, one of the things that prolactin does is regulates the length of hair. So what this mutation seems to be related to is a shortening of the hair of these animals, and that at least a shorter hair would be, associated with increased thermotolerance. And so long story short, the SLICK1 mutation is one of the mutations that is that has been described for the prolactin receptor gene, and that it, among other things, causes a shorter hair length, and so shorter hair will translate into more thermotolerance. We are looking into other potential things that could be involved in the effect of prolactin in these animals.
So that's a mutation that, can you see it from the outside? So can I look into an animal and can have an idea if it is SLICK1 mutated or not?
Yes, in fact, you can. So the hair coat in these animals is quite short. You can actually see that. It is a little difficult sometimes because, you know, animals will change their hair coats different seasons and different stages of their lives, right? So it might be that sometimes you will see an animal with short hair and say, oh, that's a slick animal, and it's not a slick animal. But if you are looking at animals at the same stage and in the same place, same season, and one is slick, one is not, you will likely notice the difference. Yes, they have like a shinier coat, a much shorter hair length than their non-slick Holsteins.
Okay.
Not
So
only Holsteins, right? So another thing about the SLICK1 mutation is that it was first described in Senepol cattle, which is a breed of Bos Taurus cattle that have developed this mutation naturally. And so that's what was used to introduce SLICK1 into Holsteins.
Okay. So from our more dairy breed, so would Jerseys have the same or no?
They could. You have to breed animals to introduce that mutation, right? To have them. There are currently animals that have been edited to have the SLICK mutation in Jersey breed, so Jersey calves. So it's going to be, you know, if you can use. So our study, for example, was not done via gene editing, right? We did not edit embryos to create slick animals. We bred cows with semen from slick bulls, but you could also use gene editing to create this mutation, right? And that would certainly accelerate the rate of the mutation. You could use your best animals to create an embryo and then use the best embryo to create that mutation. So you would have the best embryo with the slick, which is not something that we could do via breeding so fast.
So then there is this observation that these animals with this different hair coating, they would maybe take the heat a little bit better than the other ones, and that then could be associated with improved performance during moments of heat stress. But aside from heat stress, do cows with SLICK1 gene mutation, do they perform better or they would be the same? Because there's something that people were not looking into it without the eyes of the heat stress. Is that right or no?
Yes. Let me try to answer this question without being too long, right, with it. But of course, you need to have characteristics that are advantageous, like heat stress tolerance would be great, but at the same time, of course, you don't want to decrease production or other things that you have selected animals for, right? And the one reason that I don't want to say that people are not looking into it, because of course, when you are comparing slick and non-slick animals, you're always looking at parameters such as production, reproduction, stress tolerance, and whatnot. But you have to take into account that the genetic background of these animals will be different to some extent, right? So if you're comparing a cow that is a high-producing cow with a cow that is a medium-producing cow, it's going to be very hard to compare their production. And if one is a slick and the other one's not a slick, then you would be confounded by the production that they had in the first place. But when you look at, there is a paper published actually by Dr. Hansen's group in 2014, if I'm not mistaken, where they looked at milk production of the slick animals that they had in the farm and non-slick animals in summer and winter. So they took the same animals and they looked at their production in summer season and winter season. And they saw that the slick animals had very similar production in both seasons, whereas the other animals, the wild type cows that don't have that mutation, had one level of production in winter and shorter milk production, or lower milk production, in summer. showing, okay, these animals that don't have this mutation are suffering and therefore decreasing their milk production, whereas the SLICK animals are not. So that was one of the first observations regarding lactation performance. Again, it's hard to compare on a pound-to-pound basis because animals might be different just because of other genetic variations that they have, right? But I think that this is very much something that people are looking into, and that will be looked at more as we have more animals to look at.
Okay, so then you mentioned that, okay, for you to study and making comparisons, then you say, okay, I need a bunch of these animals. So you found bulls that had that mutation, and you bred cows that had the mutation, so you could have offspring with the mutation. Is that how you did it?
Yes, that's exactly how we did it. And we took advantage of the fact that the SLICK1 allele is dominant. And so when you have one copy, you will have the phenotype. And so that had also, of course, been described before. And that's why we used bulls that were heterozygous for the mutation. Those were 2 commercially available bulls that we could buy semen from. And by using those and by knowing that our cows that are in the farms, that commercial farms that we used, do not have that mutation, we would have a similar amount of offspring that would be half sisters because they're daughters of the same sire with different cows, but they would have or not a mutation, right? So we could have, in our view, a really good control group, or probably the best control group we could have, which is animals that are more similar with each other compared to the general population because they're daughters of the same sire, but they differ in the presence or absence of the mutation, and therefore they were SLICK or non-SLICK half-sisters.
Wow, that's great. Yeah, you're right. Then it creates a very interesting pairwise kind of comparison. And then what is the, how can I say, I'm going to say it in the wrong word, but when you did that, how many cows or sisters you got with SLICK and without SLICK? Is it like a 50-50 distribution, or you have something tended more one side than the other side?
No, it was a very much of a 50-50 distribution. In fact, we published a short communication on that, putting our data from this experiment, from this study with previous studies from Peter Hansen's lab, and others looking at the equilibrium, right? The distribution of SLICK and non-SLICK animals when you have or births when you have or not have the mutation, because of course that's something that you would be interested in knowing, correct? If the mutation in the prolactin receptor, because we know prolactin, among other things, is so important for reproduction and for during pregnancy, right? That if you had a mutation in the prolactin receptor, you could be affecting somehow fetal development, and you could have less offspring from these animals that carry the mutation. However, that's not what we saw. So that was a really good piece of evidence that we had very much a equilibrated rate of births from SLICK and non-SLICK animals from these and from previous studies as well. And that's another thing that only when you have a large enough sample size, you can really evaluate these things, right? And for a long time, we had small numbers of animals. So it was really hard to say, well, I have 5 here and 4 there. Is this different or is it not? So now we had a few hundred animals to look at, and it was really helpful.
I see. So then how easy was for you to find those two bulls that carry the mutation? And did you have like the whole available bulls to search for, or how did you get to those 2 bulls? I'm just trying to assess then later is like, if I go to a farm today, what would I expect of that SLICK1 mutation happening? You know, what would be the frequency of that happening? I have no idea. Yeah.
So it's a great question. There were not a whole lot of bulls that had the mutation when we started this. We started buying the semen was 2018. There was this one bull from ST Genetics that was commercially available, and then there was another bull that was in a smaller stud that we could get semen from. That those 2 bulls are originated from University of Florida herd. So one big problem that we had when selecting these bulls. So we wanted a heterozygous bull, not a homozygous bull. There were a couple of homozygous that could probably be obtained from Puerto Rico. But from these ones, the main concern or the main drawback that we had was that they were not as good, genetically speaking, when you look at net merit, milk yield, things like that. They were not up there with other bulls. And that was particularly problematic for the dairies that we were collaborating in California, because they really look at those things a lot, and they're constantly changing what they're doing to stay on top of the genetic merit of the animals. And our bulls were just not there. And so we had to convince these people that this was an important project to do, and if they could, sacrifice some of their pregnancies to produce these animals, even though when they become cows, they would be probably on the lower range of production, because it was really important for us to see whether we could have an advantage on thermotolerance on these animals.
Okay,
but
so
it wasn't too hard to find. These animals are available, and now there are more bulls that are better, much better genetically speaking, than the ones we used. So it's a lot easier now to get a good bull that would be similar to what people would be looking for, and have this mutation. So you could would be much easier to introduce the mutation in the herds. That being said, I really don't think that there is that there is a high frequency of the SLICK semen being used yet. I could be wrong on that. I'm sure it varies according to where you go and how bad of a heat stress situation different places have, right?
Mm-hmm.
But I suspect it will probably increase as more evidence comes out, and if the evidence is favorable.
So for example, for you to identify that mutation, how did you get there? So you do just normal DNA testing like we have commercially available and it shows up there, or is something special that you had to develop? Or can you go to a bull catalog? Can you find, oh yes, is SLICK1 gene mutation, yes or no?
Yeah, a lot of AI companies now have that too, incorporated into their catalog, so that it will be in the sire. So if you look for SLICK Holstein sires, I should have done that before this interview, so I could give you a good number. But you can find that. For us to identify animals, we do genotyping, and we look for the mutation.
Okay.
So you can do that with PCR as well, and just identify the mutation.
So you do that in-house, or you send that to the Holstein Association and they get back to you, or just do it in-house?
So we did that via collaboration that we had with Zoetis. So we don't have an in-house assay, but the Hansen lab does. So they do routinely do PCR to check for the mutation. But
if you're using something like Clarified, that will give you the answer that you need as well to figure it out. So right now, if you are a producer and you're testing your animals via Clarified, I don't know if they are releasing that information at this point. I think they were testing for it to see how reliable it will be and all of that. And also because it's not that prevalent in the population, I'm not sure how relevant it was to include. So I don't know if it's commercially available via Clarified at this point. But that's not to say it couldn't be incorporated in the future. Okay. So we know how you found the animals. We know they have a different hair coating. We know the linkage from SLICK1 with prolactin and having all this major effect in fertility and performance. And then now we have the animals, and now we need to put into the test, right? So you needed a heat stress environment. And it seems like in this paper, you not only found one, but you found two environments where that heat stress would be existing into those cows. So you created those cows in those different places. So we are talking about California heat stress versus Florida heat stress. Or you produce these animals in one place and then ship them to another region of the country. That's pretty much the opposite size of the country, right? Yeah, that's correct. So no, we produced them in both places. So we had collaborating farms in both states, and we gave them the semen, asked them to breed animals with the two sires within the same week
try to balance the characteristics of the cow, stage of lactation, parity, and to the best of their ability, they did that. Because we were asking them to breed a lot of their cows, so we could get as many animals as we could get, so we could study them. We were not too picky in saying, well, only second lactation cows at 60 days in milk or at 100 days in milk. But the results came back really balanced, and so we were happy to see that. So a lot of cows being bred in different farms, and then we followed them. So we had, systematic and frequent backups that we were just looking at the pregnancies and when those cows were inseminated, when they got pregnant, confirmed with one of our sires, and when they calved. And so we followed our animals that way in all different places. And so farms in Florida, farms in California, we were monitoring all these different places to follow our animals. So once our animals were born, so we bred the cows in another way that we try to do this to increase the number of animals that we could have was to breed them in 2 seasons. Right, and that was also done with the goal of having calves born in the cool season and in the hot season. So then we could look at animals in 2 different stages of their lives, right? So we had our first group of animals be born in the cool season, so they were born between November and March, and the second group of animals was born late May, June, early July. So those animals were very young calves before weaning, so they were all before 2 months of age when we did the heat stress experiments. And well, the first group of calves were older; they were post-weaned, 7 to 8 months of age, 5 to 8 months of age, really. So then we could look at those 2 groups and try to evaluate how they were responding to the same environment, right?
Mm-hmm. So how many cows in total did you have involved in the trial?
A lot of cows. I honestly don't have that number off the top of my head. But between cows being inseminated that never got pregnant, cows that got pregnant and lost, and cows that ended up calving and giving birth to one of our calves, we had a lot of cows being bred.
Okay.
The number of animals that we ended up studying in this heat stress experiment was 204 animals.
Okay.
And so we had approximately 100 of each, SLICK and non-SLICK, in all these different farms and different environments. And then the goal here was really twofold, right? One was to try to extend our knowledge about the SLICK mutation, because of the previous studies that have been done up to now were always looking at lactating cows. And of course, lactating cows, we really care about them, right? They're making the milk that we use to sell, and the whole industry evolves around that. But also it's important for us to know if the things that we observe in the dairy cows were also present in early on, or if this is a thing that we will only see in cows. So we wanted to extend those findings and see if we would see the same effect on young animals, because we also know that although cows are more metabolically challenged and more sensitive to heat stress, and that has been shown repeatedly, we know that young animals are also sensitive to heat stress, right? And even if they can be a little bit more resistant when you put a calf out there in the sun without shade, without cooling, it will suffer, right? And there is more and more evidence coming out about that. So we wanted to know if the SLICK was also going to be an advantage for these animals. And the second thing that we were really interested in knowing was also, because of what was already known, that the SLICK mutation seems to be advantageous when the animals are exposed to humid heat stress, right? So the conditions that we find in Florida, think about Florida, everybody thinks about humidity, heat and humidity, right? So studies that were done in Puerto Rico, in other places of Central America and South America and Florida are all humid heat stress. And California is a place where animals have heat stress. The heat stress is a big concern. However, it's a very dry heat, right? So we have higher temperatures, but humidity is much lower than in Florida and other places where tropical heat is the case. So we wanted to know also if we would see this advantage in dry heat versus humid heat. So in other words, if you could extend the findings to animals that are heat stressed under dry conditions.
Okay, so you had 3 commercial farms where they have those heifer calves in California, and you had 2 farms where you had all those 100 animals. So you also had this, you call the wild type, right? That'll be the non-mutated without the SLICK genes. Okay.
Correct.
And then what would you say are the main findings of the paper? So it seems like they behave. And one thing that I really enjoy reading the paper is that there is so much to bring control features to your findings. And maybe it's related to all the research done before being very exclusively to one place or one thing. Like you brought the pairs you just mentioned, right? Hey, the control cows were sisters with the mutated, so super controlled. Then you go out there, you find different farms within the same state. So it's not just one farm, right? So you bring a lot of layers of, accounting for a lot of variability. But it seems like the location matter in this paper, right?
Yeah, and yeah, you pointed out these aspects of the design, and I will tell you that it's a plus and a con too, right? Because it adds a lot of complexity to the study, and the different management that you might find in different places, decisions that people make with the animals. We don't really have a whole lot of control over that. We really wanted to do something that was applicable and that we could extrapolate to real farms, and that's why we decided to spread out the study like that. But it also has its challenges, right? So the main findings of this study were a few. Let me see. First, looking at the animals, regardless of whether they had a mutation or not, the calves seem to be more stressed in Florida than they were in California during the period of testing. Right. And I will say that and mention that so the week we were here testing the animals, we didn't expose them to controlled heat stress, right? We didn't use blankets. We didn't put them in an environment. We left them out there, and we went there during summer, and we tested them. So it was really whatever they were experiencing in their environment when we tested them. 3 weeks ago, we had a 10-day heat wave that was above 110 Fahrenheit every day. And so I was actually thinking about my animals out there, and I was like, if I could go down to one of the farms and test to see if my animals how they're dealing with this, it would be great, right? We unfortunately didn't do that. But we do experience severe heat stress here. During the study, however, we saw that our animals were less, or seemed to be less, heat stressed in California than they were in Florida. A couple of important differences between the two places, when we measured, we had those environmental data loggers that we used just to collect relative humidity and temperature over time, and we kept them out there where the animals are for extended periods of time. We collected measurements every 15 minutes to really draw a distribution of what the temperature and humidity were during the period. And when you look at that in the paper, we have a nice graph of that. Florida is like heat stress levels throughout the time that we looked at. And so they don't have relief at night, so they don't get down to that lower THI of 65 or less, where the animals actually can breathe and recover for the next day, which we did see in California. And even in the peak of THI, which is the temperature humidity index of California, we did never get to the point where it was in Florida. So animals were much more challenged in Florida than in California. That's number one. Second, we did see a difference between SLICK and non-SLICK calves in Florida. So they were about 0.4 degrees Celsius cooler rectal temperature, this is, compared to the non-SLICK half-sisters. And that might sound like a small difference, right? But we're talking here the difference between an animal that is 39.5 and 40 degrees Celsius, or 103 and 104 Fahrenheit, right? So it sounds like a small number, but it is a biologically relevant change. In California, we did not see a difference. So one thing is our animals were all within what we would call probably the upper limit of the normal temperature range, but we did not see that SLICK animals were cooler than non-SLICK animals. So one of the things we were thinking about there is maybe these animals didn't reach a level where the SLICK would just not keep going up and the non-SLICK keep going up, or it could be something else, right? So we don't really have the answer as to why the California animals did not respond as much, but we do know that the Florida animals were more challenged than the California animals were. We also measured respiration rate in these animals, skin temperature and sweating rate. And again, we did not see major differences there. Another thing that is worth mentioning here is California calves in general sweated more than Florida calves. So they had higher sweating rates. And that's expected because it's dry heat. Of course, these animals can sweat more. They have the ability to release heat that way. And that certainly contributed to them being cooler in California than they were in Florida. Sweating rate was significantly higher in California than in Florida.
That was something that I was going to ask you when you mentioned, the animals were more stressed. So one of the points is the THI, but then you have assessment, or you made measurements into the animal. So I'm curious, how did you measure the sweat intensity? How do you call that?
The sweating rate. So we use a device that measures the rate of evaporation of water from the skin, from the surface. And so that's called a vapometer. That's what the
vapometer.
Vapometer or vapometer, you could say.
Okay.
So really just measures the speed of water leaving the skin as you're measuring. So you have a very small chamber and you just put that in contact with the skin of the animal and wait until it beeps. It's about 10 seconds, 15 seconds. And then you get that measurement, which is really a rate of evaporation over the time that you measured it for.
Do you have I'll ask you for a link for that little machine so we can show people that want to check what it is. But,
sure, it's a little machine, but it's not a cheap machine.
Oh, it's not a cheap machine.
It is what it is, though. But it's a really, really good way to measure sweating rate. There are other ways, right? This is the way we use. This is the way the Hansen lab measured sweating rate before. And so we had this because this is a collaboration between my lab and Peter Hansen, right? So we were doing this together. And we also measured skin temperature. So that we measured by infrared thermometer, just measuring the skin temperature in the skin. And then, of course, rectal temperature by rectal thermometer. We went out there and we measured all of the animals once a day for this comparative study. We also have some follow-up studies that we did in California, because in California, we were able to measure the animals more than once per day. So we were able to see what they were doing as the day is getting hotter, from the morning, which is really cool and nice for the animals, all the way to late afternoon when it's really hot. So, but for this experiment, for this paper, we compared Florida and California only once a day at the time where it was hottest during that day for the animals.
I see. So that was kind of what I can see here, right, in the paper, the measurements of respiration. How did you measure the respiration rate? Just by looking at? Yeah,
we just counted plank movements. Yeah, that was just that's the least how can I say, accurate measurement that we have, right? Because it's just challenging sometimes with the animals moving and things like that. We're always really trying to go and measure respiratory rate, count the breaths per minute before everything else. So somebody would go ahead of everyone else to count that, and then we would come and measure the other parameters as to not affect the animal. But it's the more challenging one. We did not see differences there between our animals. Except for some of the older animals in Florida had higher respiration rates than everyone else, but it was not a difference between SLICK and non-SLICK animals. So another finding that we had in the paper is we had THI in our statistical models, right? And so when we had a THI have an effect that we called either a tendency or a significant effect, we did a regression analysis between our THI and the parameter that it was showing significance. And when we looked at that by rectal temperature, so THI by rectal temperature, we were interested in the results of that analysis because it showed us that for non-SLICK animals, there was a linear increase in the rectal temperature of the animals as THI went up, right? So just comparing the two, THI goes up, there is a correlation with rectal temperature. And that did not seem the same for SLICK animals. So from that analysis, it seemed to us that SLICK animals were able to stay a little bit more constant as the THI went up compared to non-SLICK animals, where you have a much steeper line of correlation. So that was an interesting finding to us, was a different way of looking at the same data. And of course, we need to follow up on those things to see exactly what it really means.
I see. And yeah, I mean, there's so many other things, right, that you can think about it. But another thing that you could think about that environment is that, do you think as a source of heat, like, for example, whatever the cow was eating, did you see any huge differences, or cows were on pasture in Florida versus they were confined in California, or that any discussion on, hey, we are feeding a bunch of bad hay that most likely could increase a little bit of their temperature just by fermenting versus, California, where you have maybe, or the other way around, and how this SLICK gene would also interact with that, right?
Yeah, that's a great point, Phil. We didn't look into that, right? That's not something we considered, but certainly something to think about. You're right. So we didn't account for what the animals were eating as a source of potential heat, for sure.
I see. But you do have variation on the place, right? So
for sure,
not only humidity, that may be more direct, but you may have some other indirect things, like you said, complicates a little bit, but makes it easier.
Yeah, but I mean, it's building blocks, right? So I think that we have the results of the current experiment, and we're following up on that. And hopefully other people will also be interested in looking at other things related to this mutation and other ways to decrease the impact of heat stress. And then, together we can build a better understanding of it. There's just a lot of variation that it's really hard to account for. If you do a very controlled study, typically you're going to have a very limited set of conditions, right? And then to extrapolate that to another set of conditions, it becomes challenging as well. So, I mean, you would always have that balance to work with. But we are, just mentioning the following up, since these animals are still in the farms, we are following up with them, right? So we had this experiment done. That was back in 2020, in the summer of 2020, in the middle of the pandemic. We were out there wearing masks and measuring these parameters in the animals. But they stayed in the farm, right? So we had a chance to collect data to look at reproductive performance. And now that they are becoming lactating cows, we are also trying to evaluate their lactation performance. So there's going to be more coming out, hopefully, as we get more data from these animals.
That's great. So out of the 100, you have 60 per treatment, roughly. Or you're saying out of the 200, you have 60?
No, we have, well, for this study, we had 200 animals. We likely don't have the 200 anymore because animals get cold from farms, and that's not something that we can control, unfortunately. But we are following up on the animals that we still have to gather as much data that we can.
So if I want to, it's almost like you're doing like a, let's say if I want to do a trial today with SLICK1 gene mutation, I took you a few years to build that cohort of animals, right?
Yeah.
So there's no way you can expedite that in the way you did. If you want to do another trial, would you do the same way?
Well, I mean, you would still have to produce the animals unless you can find them out there, right? So if the farms are using the SLICK as one of their desired characteristics that they want to introduce into their herds, then you will start finding animals that have this allele. and that have the SLICK mutation phenotype. But unless you have that, you would have to produce the animals, right? So you could either do that by breeding cows or by gene editing embryos, but you would still have to put them in a cow and then have the cow gestate and have the animal be born. So it will be hard to get around that if you want to produce more animals to study. So that's one of the downsides of the cow because of the long generational interval, but that's our species of interest and we want to learn about the cow. So that's the constraints we have to work with, right? But you do have to produce these animals. Now, one of the things that I will mention is that since the animals that we have out there have one allele of the mutation, right? They could and they will pass that along, right? So you would expect to see animals, offspring in descendants of those animals to still have the mutation at a lower rate, perhaps if the sires they're being bred to don't have the mutation, but you would continue to see that.
Okay. So one thing I found interesting is that when you did the skin temperature, you have a clipped versus an unclipped. And
Yes.
it seems like the direction goes so I found that curious. Why did you decide to clip? Because I would think you just do something without even clipping the hair.
Well,
I guess?
so the clipping meaning removing the hair, right? So we wanted to have an area of the skin that didn't have any hair. So if there was an effect of hair, we would be able to see it. So we measured it in the skin covered by hair and in the skin right next to it that was clipped or didn't have any hair. So we wanted to see, okay, is this skin temperature because of hair or because of the skin itself, right? And we did that for both the sweating rate and the skin temperature, so we could compare clipped versus unclipped. And that was another place where we found a small difference between our SLICK and non-SLICK animals, specifically the young calves in California, where clipping the skin did not seem, or clipping the hair off the skin, did not seem to change their sweating rate as much as it did change for non-SLICK. So in other words, a wild type animal or an animal that does not have this mutation, if you measure the sweating rate in the skin that is covered by hair, and then you measure it in the next spot where there is no hair, there was more sweating where there is no hair, right? Which is expected. So the hair is keeping the water from evaporating. Where you don't have hair, it evaporates easier. In the SLICK animals, we did not see that difference, meaning that the hair is not really in the way of these animals sweating, which is on itself an advantage, right? So lots of findings there that we would love to follow up, do a second study, see more animals. Really, this is all about repeating, right? And being able to see these effects over and over again to really have confidence that yes, this is something that we can use in our advantage and in the animal's advantage to decrease heat stress. Of course, I will say, right, there is not one solution. So the SLICK1 allele might be one of the ways that we have, one of the tools in the toolbox, but it's really not going to be the one thing that's going to make a difference, right? So everything together, like cooling management and other things that we use already with the SLICK mutation, perhaps, is what we need. But I wish we could just have one solution that will take care of the problem, but usually
would
not.
you, after this work and some of the other work that maybe is coming, would you think that's something that farmers should be considering, to be selecting for bulls with the mutation, with this SLICK1 mutation?
I honestly think that given the data that we have to date, that does not show any detrimental effects of having this mutation, and with the advantages, even if small, I would say it's certainly something interesting to consider. If you are in an area, if the farmer is in an area where heat stress is a problem, I would certainly consider that as one of the things I would want to include in my selection strategies. Why not, right? Unless we find a major problem, but we haven't. Everything that has been looked at, it seems like the SLICK is not really affecting production, reproduction, performance of the animals. So we are, I will say, following up on some of this to try to understand the cellular effects of this mutation, and we started by looking at the skin. So we went out there and we collected skin biopsies of the SLICK and non-SLICK animals to try to understand better. So what is it doing to increase thermotolerance, right? Because the results in sweating rates, so it seemed that maybe the SLICK animals would be more able to sweat, so they sweated more, and that's why they were cooler. That's not always the case. We didn't see that in our animals, for example. So it's still a little bit unclear what makes these animals more thermotolerant. And so we're trying to increase our level of detail in what we are trying to find to see if we can understand a little bit more about prolactin signaling and how it's different between SLICK and non-SLICK animals. So it's really trying to get at the, biology behind this, what we are observing, right? The increased thermotolerance. So what causes it?
Okay, very interesting. Well, you were very generous with your time to talk to us about this. Any final considerations that you have on your recently published paper?
Just hope everybody enjoys reading it, and that it helps us better understand the roles of this mutation. So I mean, I think this is very exciting research. I was really excited to be part of it. So
it seems like all the Holstein farmers, they were excited because there's a component from the Holstein Association here, isn't it?
Yeah, certainly. This is funded by the Holstein Association USA, right? So that was really why we were able to do these big studies, because they provided us the 3 years of funding for it. So certainly, and that shows that this is something that the Holstein Association is seeing as an interesting area to study. I was recently at the Holstein convention giving a talk about our main findings, and it was really well received. So people seem to be very interested. So we'll see where this goes. I do think that there is going to be more research done in this area to understand to what extent the benefits of this mutation are to thermotolerance. So stay tuned and let's see what else comes out from both my lab and other labs studying this.
Okay. Thank you very much, Anna. Have a good one.
Thank you, Phil. Thank you for giving me this opportunity. It was great.